Hey, Cheryl, are you doing the introduction today?
I can certainly do the
So I
introduction.
think you obviously and Mehwish, I'm looking forward very much to your presentation, and I'm really excited to see your great career progress. And this is a celebration of that. But I think it'd be nice, if possible, if Cheryl was willing to do this.
I would be absolutely honored to give the introduction. So why don't we get going? I know that there's some people that are coming on, but it is such an honor and a privilege to introduce Dr. Mehwish Anwer today as part of our trainee showcases at Pathology Rounds. Mehwish has been working with me as a postdoc since 2021. She obtained her PhD in Finland in 2020 and received both her Master's of Science and Master's of Philosophy degrees in Pakistan at the Lahore University. She has no less than 33 awards to her name, and is recognized in both for her research scholarship as well as her outstanding mentorship and engagement activities. To give you an example, in 2023 alone, she won the Fast Track Diversity Award to attend the Bright Focus workshop on Alzheimer's disease. She was awarded by the Society for Neuroscience, won the 3M Slam competition, received the Bluma-Tischler postdoctoral award, a travel award from the Canadian Traumatic Brain Injury Research Consortium, a pathology and laboratory medicine mentorship award, and the Best Presentation Award from the Faculty of Medicine for the futures of, I forget what the actual title is, Mehwish. She has 15 publications and 2 under review, several from her postdoctoral work so far, and has given 32 presentations in her career. So it is really wonderful to see such a promising young scientist develop a CV package and the foundation of what I will expect to be a very long-lived and very innovative aspect to reimagining how we do neuropathological experiments in animal models. So with that, I hope you enjoy her presentation, and Mehwish, please do take it away.
Thank you so much, Cheryl, for this lovely introduction. It's my absolute honor and privilege to be a part of the Pathology Round session today. And thank you, everyone, for joining and listening to what I have to share with you today. And I will fire up my screen and go through a little bit the objectives of our talk today. And can someone just confirm that you can actually see my slides?
Yep, all good.
Yep, very nice.
Okay. Perfect. Thank you so much, Cheryl and David. All right, so the talk that I will be giving today is a snapshot of the work that I've been doing with Cheryl, and as part of the greater community of pathology and laboratory medicine department at UBC Vancouver, as well as being a member of the Javad Mufagian Center for Brain Health. And this has enabled specifically because of the excellent mentorship available to me and the resources and facilities to develop my research career further. And it's an absolute privilege to be a postdoc fellow within this department and institute. I will be talking a little bit about the background, why the research that I'm conducting is important, specifically in the light of traumatic brain injury research and the need to do more in the field. I would also be introducing the 3D labeling methods, the 3D histopathology concept. We had a little bit of snippet of that in our Pathology Day 2022, and this is really synergistic with those efforts. But the exciting bit is that we have these setups now locally, and we are actually leading on these fronts internationally as well. Briefly, at the end of the talk, I would also introduce another approach, which is spatial transcriptomics, and would love an opportunity to come back probably next year and put that into much more detail to fit in with the time constraints we have today. So moving on to the first part of the talk is, while it is very well established and known that traumatic brain injury is a very complex disorder and concussion is a big challenge, it is still not emphasized and budgeted enough in federal fundings to address this need. And just a snippet of our local situation, that TBI still remains a huge burden for the people of British Columbia as well as greater community in Canada. And within this scope, there is billions of dollars of investment that is required to kind of deal with the direct and indirect cost of brain injury. And there was a very extensive report back in 2020 from the Public Health Agency of Canada that emphasizes on the needs to address this challenge, which is essentially an acquired injury to the head, either by an external force or through another neurological insert that could result in disruption of the normal function of the brain. Now, given this dire need to address this problem, it is also very important to understand that traumatic brain injury is not an isolated event, and it does not represent just one pathology, but is actually resultant and a risk factor for many other comorbidities, and including the development of post-traumatic stress disorder, as well as dementias, including Alzheimer's disease and related dementias. Patients or people who actually experience brain injury and concussion complain of multiple symptoms, including depression, anxiety, insomnia, fatigue, and other neuropsychological impairments that overlap with these associated comorbidities, which in turn magnifies the impact that brain injuries can have on the health system as well as on lives of the lived experience people. Now, it makes all the sense when you start looking at the neurohistopathological aspects of what traumatic brain injury does to your brain. And it is not a single isolated event. Instead, it is a myriad of processes that are initiated upon trauma. And depending on the severity and the nature of the injury, there can be multiple cascades at the molecular level that are initiated. This could include damage to the axonal bodies, the fibers that connect the brain, as well as damage to the vascular components of the brain, which is essentially the circulatory system and the way the brain gets its nutrients and oxygen supply. As well as there are damages to the integrity of the neuronal populations, the glial populations, and the neuroinflammatory component of the brain upon traumatic brain injury. Now, all of these events that are unfolding at different levels and time windows after traumatic brain injury could actually result in further complications, including and not limited to post-traumatic epilepsy, dementia, vascular diseases, neuroendocrinology diseases, and beyond that. What does this tell us to a preclinical researcher like me, who essentially do not engage with patients and directly, but have this huge problem at hand to understand what exactly is going out in the brain, and how can we really address and mitigate at least some of these processes to understand the evolution. But then there is a huge challenge. No two brain injuries are same in terms of how they happen, in terms of what extent the damage was, and also in terms of what are the outcomes for the people who underwent those injuries. Furthermore, to add to the challenge, the diffuse brain injuries that are much more concussion-like are harder to pinpoint. When you have a car accident related brain injury, it's much easier to pinpoint the location and the severity because of the visible damage to the skull as well as the underlying brain tissue. But for these diffuse brain injuries, it's almost like a needle in the haystack to identify where things went wrong. And these injuries do not show up on CT scans and many of the blood biomarker, if any, assessments that we do thus far in clinics. So this leaves with a question for us in terms of modeling of brain injury, is to understand the brain-wide landscape of what is changing in the brain after traumatic brain injury, especially the levels that are much more milder or concussive. So my research activities are surrounded around this big question, is to understand and address this unmet need of understanding the mechanistic details of brain injury, how does this affect, and then to be informed and discuss back with the clinical community to understand exactly what can be done in identifying novel biomarkers as well as therapeutics, and to even understand the function of the biomarkers we do know thus far and how they are shaping the brain after an injury. Here, what you're looking at is a preclinical model, so an animal model that was developed by Dr. Wellington and Dr. Krypton's lab, and we're honored to have Dr. Tom Schenk also still a part of the team, who was one of the prime inventors and has been characterizing the model thus far. And it's my privilege to work with him and learn the biomechanics. This animal model essentially enables us to study that subconcussive or concussive mild injury that we're talking about. The strengths of the chimera model is that it is a non-surgical model, so it mimics much more the human milder site of injuries, where you do not have a visible focal lesion site, and it is also a free head movement model, so it mimics a lot of acceleration and rotational aspects of the brain injury that would inform us in terms of how the different underlying pathologies occur. So my work depends on the use of a chimera model. Now, there has been a lot of discussion in the international community of neurotrauma research as to how we solve this puzzle. How do we understand what is exactly happening in the temporal windows we're looking at, as well as how do we pinpoint the intersection of different pathologies? And a multimodal, multidimensional approach seemed to be the best option thus far, where we can actually incorporate studies from rodents and then kind of trying to translate them all the way to upper, higher order organisms and then to humans eventually. But then the process is live on the spectrum of different sorts of outcomes. And these spectrums not only include the occurrence of the misfolded proteins and like tau, amyloid, but also in terms of neuronal activity of the brain, in terms of connectivity within the brain, and methodologies like tissue clearing and multidimensional imaging, which I will be talking a bit more in the second half, as well as RNA sequencing technologies enable us to understand what exactly happened at the time of trauma, or subacutely or acutely, or even at the scale of many months of the trauma. How does the cellular response change in the area that might have an impact, or the areas that are connected to the area that might have an impact? And then overlaying these information from transcriptomics level to the protein level, and then back to structure levels using MRIs and functional MRI and to electrophysiology would seem to be the only way to actually understand the links between these events and to decipher how they can be modulated for a better treatment of brain injury. So one of the things that I embarked on in terms of optimizing for my own projects is to move from conventional two-dimensional histopathology analysis in preclinical animal models to a bit more of a three-dimensional approach. Now, the rationale behind this is while the two-dimensional histopathology has informed decades of research work and actually laid the foundation for what we are studying now in neurotrauma and beyond, it has some limitations to it, which were essentially guided through lack of technology at the time. And something that is very crucial to understand in this aspect is, especially for mechanistic insights from brain damage, it is important to understand that the structures that make a brain the way it is are complex and three-dimensional in their arrangement. So if we are looking at a few planes or sections from a brain, based on our understanding of the injury thus far, we might be actually overestimating or underestimating some of these changes and might be undersampling for the distribution and the complexity of these structures. So it would be wonderful if you could actually look at the brain as a whole without doing this unbiased sampling and looking at how these things connect to each other. Having that in mind, I started with reviewing a lot of literature in terms of understanding what are the approaches that can allow me to do this whole brain methods. Now, MRI is one of the techniques that are used for in vivo understanding while the mice are still alive, which is great and fantastic to see the damage. But then the resolution that is offered by MRI does not allow us to look into cellular level details, as well as the possibility of labeling the post-mortem tissue with multiple analytical approaches is not available for MRI. So we needed a method that could allow us to do something beyond MRI imaging, and looks like that the three-dimensional tissue clearing approach offers that space. There are multiple methods that have been proposed in literature, depending really on the reaction chemistry. Some of them are more organic solvent based, and these are the early methods. Some of them are still being very popular, like the DISCO approach. And essentially these methods are designed in such a way that you do not have to cut the tissue into sections, but make it transparent or visible enough for the light to pass through and image the components within. However, some chemistries have more advantage over the other depending on how much they modify the tissue that we're looking at. So after the organic solvents, there was much more aqueous-based tissue clearing methods that were developed. And then more recently, hydrogel embedding based methods that the parent technique called Clarity, on which much of my work is also based. And then there's a lot of versions of that. The advantage of this technique is that you actually embed a hydrogel system into the tissue before you remove away the lipids from the membranes. By removing, I mean rearranging them in such a way that light passes through them. So this gives superior integrity to the tissue that you're about to actually analyze in a different way. And combining the Clarity method with another method called Shield enables it much better, and the tissue is firm enough to be manipulated with the three-dimensional microscopy approaches. In the lower panel, you can actually see some examples of the mouse brain as well as a small human brain tissue block that has been made transparent. So what I'll be referring to as clearing throughout the talk. And then how we could actually get a lot of three-dimensional information out of these tissue blocks when taken to the respective microscopic assessments. Now, how does this workflow look like? And for those of you who have been doing routine histopathology, some bits of this are very familiar and actually based on the same principles. The only difference is we do not need any cryosectioning or vibrotome sectioning or microtome sectioning for these tissues. Instead, we take the whole organ as it is, in this case, the brain tissue, and then we start delipiding it in such a way that it start becoming translucent and then eventually transparent, depending on refractive index matching in the last step. Now, this whole chunk of tissue in mouse brain, we're talking about 14 millimeter to 8 millimeters in dimensions from the rostrocaudal axis and the width of it. And this is a huge tissue. And considering any microscope that has the ability to image, cellular level details, this is impossible based on the optics we have. But then more recently, a microscope that was built based on the confocal concept, but with much more working window and field of view, which is called light sheet microscopy, was developed, and this allowed for imaging of these large chunk of tissues, and then to actually process this data such that you can identify and segment out the cells or the features that you're looking for, and then trace back these information to an atlas to identify the spatiotemporal locations of the information that you get. Now, this is really the overall flow of the work of how this is done, but many bits of the processes are still really in their infancy, and then there's a lot of need for especially the second half of this workflow, which is to analyze the data, because using the big data tools and machine learning approaches that are available to us now, this is now getting much more possible and wasn't the case 10 years ago. So let's dive in a bit into all these individual steps of the process. And this is the example that I briefly mentioned is Shield, and that's what we are using for the tissues that are being processed in Wellington Lab. And Shield is essentially an additional layer to Clarity. So in addition to the hydrogel mix that we will incorporate in the tissue, we also use some epoxide linkages to make a bond between the tissue to give it much more stability before we clear it. And the tissue quality that is generated based on this method is very, very good. And I can tell you from my experience now that the tissues that I prepared back in 21 are still good to image in 24. So the longevity of the tissue is really enhanced by doing this additional step called Shield. This was actually developed by the same group who developed Clarity long ago, and it's led by Dr. Casey Shang at MIT. This is an example of some of the tissues we process in our lab, and you can see in the left panel is one hemisphere of the mouse brain, and the first image actually shows you how it looks like before anything is done, right after being harvested from the skull. But then over the time of about 2 to 4 weeks, you start seeing the tissue becoming transparent and the grid becomes much more visible. On the right side, you have an example of a whole brain that was recently cleared, and you can appreciate how it has become uniformly transparent for us to go through it with the microscope that I'll be showing you now. So one would wonder that what is different in light sheet microscopes compared to confocal microscopes that enable us to do this? Well, while there are differences in sample holders in terms of placement of tissue and how it is imaged, the key element that enables us to actually use this microscope for large tissue imaging is the fact that the sample is not illuminated all at once, which is the case in confocal. Instead, there are two beams on the left and the right of the tissue, and these beams intersect such that only a small portion of the tissue is illuminated at one point, captured, and then the beam moves further or the tissue moves, depending on which models we're using. Now, this really allows us to image huge chunks and volumes of tissue without actually bleaching it because of constant exposure by the lasers that we are using. So this really enabled the whole brain imaging paradigm that we are using now. This is just a snapshot of the microscope we have at UBC, and thanks to Dr. Fabio Rossi at BRC Vancouver campus, who has allowed us to use his microscope. And this is one of the favorite spaces I have on campus, where I spend sometimes hours imaging these beautiful neurons and cells that I'll also share with you today. And looking at the right side of the slides here, which is actually a snapshot of how things are arranged inside the microscope. And this is exactly what I just stated, is there are two beams and there's an optics or objective at the back, and the sample is placed in the middle to be able to capture it properly. Now, there are some considerations like the size of the chamber that we will put inside to hold the tissue. It has to have enough field of view and working distance to allow imaging. And then also the tissue should be rightly index matched so that the light can pass through the solution in which it is sitting as well as through the tissue, so be able to image it without any shadows or diffusion. We have also worked with developing, we've actually bought a chamber that is modified to accommodate the whole brain. And this is by Translucent Biosystems, who are also an active partner in helping us optimize these technologies. As you can appreciate, there's a lot of technological aspect as well to this before we are actually enabled to look into the changes of these brains. But then we are very happy that we're at a point where we can use the system to address many, many questions now. Just an example for you to enjoy here, these little gummy bear brains that we get after tissue clearing. The left is an example of a little hemi mouse brain that is tagged to the holder in which it will be imaged. And the right side, you see a full whole brain with a bit of a spinal cord also attached to it. Now, this work, the basis of the setup of the whole technology with an example of brain injury looking at neuronal activity, we have uploaded the manuscript reporting this work at bioRxives last year, and we're preparing now a very extensive manuscript to be submitted soon. So if you're interested, please go check it out. And in this particular study, the method and approach that we use was to look at neuronal activity changes after TBI, because this is really essential. We do know that we have cognitive abnormalities after brain injury, and then we have functional abnormalities. But then how does that look like from a standpoint of early immediate gene reactions such as CFOS? Fortunately, with mice work, we have transgenic mouse lines that enable us to look into these activities in real time, much real time. And the mouse that I use here are called CFOS trap. So upon TBI, all the neurons that responded to an event in terms of activity within a small time window after brain injury, they are locked in such that they will constitutively glow when we harvest the tissue by injecting tamoxifen. And an example here you can see is a snapshot from a cleared brain that is showing CFOS activity in these neurons of the cortex. And you can appreciate the resolution that we were able to optimize to actually get it really clean cellular details. This is a snapshot of how the study design looked like, and we were able to capture the spatiotemporal aspect of this brain injury by looking at the CFOS expression throughout the brain. And you can see in the panels that we start from all the way from the dorsal most part to the ventral most part, or we could also have a sagittal view. So this imaging approach enables you to go back and investigate the tissue in any view or aspect you would like, because essentially you have captured the whole brain as it is with the labels you want, and then the tissue is also preserved for multiple labelings after that. Just some more examples in terms of the quality that we were able to achieve with this method, and to be able to look back at these sections and images. Here I prepared a small video for you to see how it looks like when we do 3D reconstruction of the images that we acquired. And also noteworthy mentioning is that this process of acquiring these 3D images is one challenge, but then to be able to stitch it with representative volumetric details and also have the resolution intact took us quite some time to figure it all out. And using specialized imaging analysis approach, we are now able to capture it, 3D render it, and also analyze it for the pathological changes. So once we had the whole brain image, the next question was, how are we going to analyze this data, right? So it should be built on the principles of feature extraction and data segmentation to be able to see how many cells, for example, were activated in hypothalamus or hippocampus or cortex after traumatic brain injury. But then to do that, we have magnified the scale of our assessment many folds. Having a whole brain means we need to have geographical landmarks inside the brain to actually go back and map and identify where we are. So while we solved one problem, we created another one to be able to assess this. But thankfully, there are tools out there, and we worked with in developing some of the open access toolkit, which is called BrainCon 3D CFOS. So it's built on a already available package such that it can process the images into tangible data images that we are used to dealing with previously, and then develop an elastic filter that is capable through machine learning to actually identify cells from background, and then use these cell coordinates and map it to the reference atlases. Now, the mapping part to the atlas is really, really new, and there's a lot of literature out there, but we were able to optimize this such that your mouse brain atlases that sit on the lab bench with all the maps of the brain are now 3-dimensional and digital, and you can superimpose those maps on top of our whole brain images. We just for reference, for each hemisphere of the brain, we were able to identify 1,600 regions that gave us cell counts of the CFOS in this example. Now, this is huge data, and to be able to make some sense out of this data and to visualize it with dimensionality reduction, we also developed multiple scripts and pipelines to create these plates of information to map back the CFOS activity and understand which are the areas that change within TBI group compared to the sham, and also created differential maps to look at those areas. We also developed some scripts in R to be able to dissect out these numbers as needed. And one of these examples you're looking at is called a sunburst plot. And these sunburst plots they're interactive, and then you can actually go to each region that you care for and look further into the details of what you are looking. For instance, in hippocampus, if you want to look into CA1 layer, you can click further and get the counts of those regions. So really enabling you to go throughout the brain and identify the changes you care for. This is also work in progress. By getting CFOS activity, which is a surrogate of neuronal activity in the brain after TBI, we were also able to do some predictive modeling in terms of understanding how these circuits are changed and which all functional networks are affected by changes in CFOS. And we actually borrowed the network information from MRI data sets from both human as well as mice work to see what is happening at level of CFOS activity. Here's just another example how you could also validate this information. Now, when we began this work, many a days we were wondering, we are dealing with a black box, when we depend on codes, it's very different compared to looking under the microscope. So we also conducted heavy validation studies to recapitulate what we see here using either 3-dimensional ROI drawings and quantification, as well as doing conventional 2-dimensional. So we took out some planes as if they were sections from a brain, and we did those analysis to validate the accuracy of the method. And this is a depiction of how you could actually isolate a 3-dimensional ROI and identify the objects, in this case CFOS cells, within that region and look at them comparing between different disease states. In this case scenario, we are looking at brain injury, but essentially these methods are applicable to any preclinical modeling for other neurological disorders as well. Here is another approach we recently optimized, and this is what you're looking at is an atlas overlaid over a brain image. And for the first time, we are also able to now interactively do this process. Previous, the method that we reported in the preprint article that I mentioned was based on a Python code, and then you get the resultant output in terms of CSV sheets where you look at the numbers in each region. But now we are also able to do this much more interactively, which gives us even more confidence and selective power in looking at subregions that we find important or have changes within. This work was being carried out in collaboration with Zeiss, and shifting the analytes that we're looking at, I also want to highlight the work of our master student Anwer, who I've been a mentor to for her last 2 years within the lab, and she did spectacular work in optimizing to label whole brain vascular imaging, and we were able to identify unique sites in the brain that were not gross histologically as evident to show blood-brain barrier damages using this approach. And the resolution here is also beautiful in terms of how much details we can reconstruct. And it is much more appreciative in this scenario when you think about the utility of this approach, because vessels are much more tubular and three-dimensional structures. And what we see on a two-dimensional plate is usually a circle or an oval. But with this approach, you're actually able to look them in their native morphology. Such is the case with the axonal damage, and this is a work I have been conducting in collaboration with people at ICORD who are interested in also looking at spinal cord injury element. And we are also using these specific transgenic mice lines to understand what happens if the brain is injured from one side versus the other, so a lateral versus sagittal impacts. And these models allow us to look at all the fiber bundles and to predict and understand which all connective pathways might be affected now that we have a map of the entire axonal segment. Using machine learning, one of our co-op students, McKenna, is actually doing a great work in identifying axonal damages from these large data sets. And as an example on the right, where she was able to train the model to actually pick up those axonal damages from this highly dimensional data set and to pick up details of changes in axons from different levels and sizes of axonal bundles. So this is a work in progress, and we are very excited about the utility of this as well. More recently, with help from Antoine, who's a master student visiting us from France, is also looking at the automated methods to quantify amyloid plaque pathology as well as cerebral plaque labeling from the brains that I prepared for him, and he's developing the 3D code to actually segment these out and enhance our understanding of how these two things interact with each other after trauma, or also in case of Alzheimer's disease animal modeling. Before I conclude the conversation, I also wanted to highlight a recent example that actually excited us much more from human postmortem tissue. And I think the utility of this approach, especially to understand underlying pathologies, is really enhanced. And in this recent study that looked at what happened to a postmortem tissue upon a treatment for Alzheimer's disease, was really remarkable. If you look at the down video, which is in green and orange, this is actually a vessel from a postmortem human brain, in which orange is the amyloid deposition in that vessel. And it is really beautiful in terms of how much detail now we can extract from postmortem human tissue using these approaches. Right, so that was a bit of the work that we are doing on the side of brain clearing and three-dimensional imaging. Briefly also touching upon our work that we are doing in collaboration with Dr. Mark Simbrowski's lab at Life Science Institute, UBC, which is essentially enabling us to add one more layer of spatially enriched data to what we are looking at. And spatial transcriptomics is one of the really shining and emerging methods. Classically, we look at the transcriptomics or changes in gene expression using single-cell RNA sequencing or bulk RNA sequencing from tissue homogenates or from blood samples. But what spatial transcriptomic enables you is to actually look at the gene expression changes at the site where they're happening. So basically combining a histological preparation with a transcriptomic assessment or RNA-seq data. We have conducted sub-experiments using the 10x Visium genomics approach, which is allows you to collect sections from the area of interest from your brain and overlay them overlay them on a specialized slide that already has barcoded information that corresponds to transcriptomic details, and then to be able to do all things in one space. You can do immunolabeling, you can do RNA-seq analysis, and then overlay this information to see what happened and where it happened at the same time. This is an example from our own study that we did. In the first panel, you see immunofluorescence preparation of the section where you have GFAP, that is a marker for astrocytes, and DAPI for nuclear labeling. The same section underwent RNA-seq analysis using the method that I just mentioned. And the middle panel actually shows you these multiple circular areas, and each one of these circular areas are actually a complete RNA-seq data set with 30,000 gene information. So we ended up generating huge amount of data from just 4 pilot experiment brains, and now we are working towards deconstructing this huge amount of information, again using machine learning as well as advanced data interpretation and visualization approaches to see what is happening, where is it happening. In the rightmost panel, you see an example of any candidate gene that you're interested in, and it gives you right away a full picture of where all things changed. One of a really shining student in Mark's lab have actually written up a code to put a Shiny app together, and this app allows us to actually interact with the data. So many of us, like myself, we were not initially trained to actually do a lot of coding while looking at tissue histopathology and damages in transcriptomics. But now with these visual interfaces, we can actually go and interact with the data, pull out the graphs we care for, and have these RNA-seq informations overlaid on top of the immunofluorescence information, and even generate plots to see the differences between sham and TBI cases. So with that, I'd like to conclude by emphasizing a few points, and I hope I made the case here for much more collaborations together to enhance our ability to actually look into three-dimensional histopathology, and also learn from your experiences as clinical histopathologists on how we can use this further for building the connections and decoding the complex puzzle. So key take-home messages: we know TBI is a heterogeneous and complex disease, and it's very crucial to understand what is underlying these outcomes. And preclinical modeling and post-mortem tissue actually provide great avenues for mechanistic studies. So we would really like to benefit from this opportunity. And modern tools that enable spatially enriched investigations really allow us to understand the functional importance and pathological dysfunction related to these traumas. With that, I'd like to conclude and say a big thank you to my mentor, Dr. Wellington, as well as to the team that I work with in her lab, which is the best environment one could have as a trainee, and very well supported by the NINS cluster, Jeff, especially as well as all other mentors that we collaborate with in different projects. And I'm happy to discuss if this interests any one of you beyond this lecture today as well, to build some more connections and collaboration, as we are excited within Department of Pathology to do so. And open the floor for any questions you may have. Thank you very much for listening.
Thank you so much, Mehwish, for a really informative seminar and a wonderful recap of the work that you've done so far. I will open the floor for questions.
I'm sure I wasn't the only person whose kind of mind was buzzing with thoughts about how the optical clearing and three-dimensional microscopy could be used to study, other diseases of interest, of course, for someone like myself, it's in the oncology space. And just, I think I probably know the answer, but just to ask the question, would you guys be open to collaborations outside of brain research to, explore specific questions?
Absolutely. And I think we have taken some steps in that direction already by educating and training many more labs on campus. And I can tell you that I have cleared and imaged any body part available from a mouse, including arms and bones and nails and heart tissue and everything, because the method really is diverse and widely applicable to different sorts of tissues. The only thing that needs to be worked around in terms of how long you want to leave them in those reagents, and do a little bit of grading experiments to see what's the best combination. But it is widely open to different sorts of questions, and we are very excited to keep nurturing this methodology for different sorts of projects. So yes, absolutely.
We have, and actually Jonathan, who's got his hand up politely, is working with us on us. We have a mouse model which has lineage tracking attached to it. So the early lesions are all tomato positive, which presumably would be visible. And it would be really interesting to look in 3D to see the expansion of the TD tomato positive precursor cells and where they are within the kidney. And I think a kidney is kind of like a little bite-sized piece, which probably is workable with this technology, a mouse kidney.
Yes. Yeah, I think we could. Please, sir, go ahead. Yeah.
Oh,
Okay.
yeah, I don't want to like interrupt you at all, Mehwish. So please finish your thought first.
Now, I was saying that kidney tissue fits within the capacity of the microscope we had right now, because we are also learning that at some point we need different instrumentation, much more advanced, that have a bigger space to put up larger size tissues. But a mouse kidney or a rat kidney at this point should be able to fit into the chambers that we have to image that sort of signal. Yeah.
Maybe we should discuss this in terms of the CEIBH, the Odessa CFI, which may or may not go in. It could be a nice extension and tie it to MapCore as like a new functionality in pathology.
Absolutely. Yeah.
I think certainly as we especially understand, the emerging world of oncology, which David, I don't know a lot about, but certainly the spatial resolution of the different cell types in a tumor is becoming a very, very important topic as far as I understand it. And so I think these sorts of methodologies, the overlaying spatial and 3D analysis methodology will be a very important part of the future of where we're going. Jonathan, you have your hand up. Please go ahead.
Yes, thanks, Mehwish. So I'm a clinical pathologist, and so I'm used to looking at tissues under the microscope, and you're trying to take my job away from me, being helpful to Dr. Huntsman. But my question is, I had to step in and out because I got called to the lab. Did you discuss, have you tried sectioning what the tissues look like after you're clearing and optical mapping with the microscope? Like, can you then go back and look at the slides in a conventional manner in any way?
So there are adaptations to the method. So the short answer is yes. We have tried doing some of them, but what I've personally observed, it is better to have sections up to 100 micron thickness, because if you try to do the more conventional 4, 8 or 12 or 20 micron, then they are a bit fragile because we have modified, how the tissue holds itself. But then there are adaptations to the method in such that you could reverse this and actually have some sort of labelings that you care for eventually in the sections, first embedded, then clear the tissue, and then take out the areas you care for and build up sections. So there's a lot of adaptations to the method that could actually enable doing that. And one of the challenges with tissue clearing is when you generate a tissue of that large size, not all antibodies or markers are happy to run through that tissue from periphery towards the center. So more and more, the field is also trying to build up pipelines to actually get the full brain imaging, what you could get out of it, and then divide it into section-like or thick chunks of tissue of specific areas to be able to label them with markers that we know are established for the field. So definitely that's doable.
And this is a bit outside of my particular research interest, but I deal with a lot of placentas and perinatal samples, and everyone tries to take like 50 to 100 sections consecutively of a basal plate where the invasion of the placenta is going into the endometrium, and everyone wants to assess the vasculature. In theory, would this be, we wouldn't be able to do any kind of transgenic studies or something. This would be clinical samples, but do you think that 3D mapping would be possible in terms of that?
Mm-hmm. Yes, I think it should be. The only challenge with more flaccid or not so rigid structure organs like placenta that I would like to imagine, and again, not an expert, would be to add an additional layer of a mold. And we tried doing this with some of other tissues, is to embed them in agarose before doing the whole approach. And that seems to work perfectly to keep the whole length of the tissue that you're looking at in a imageable space. And then it depends on how thick the tissue is in terms of the dimensions in X, Y, and Z. Even if it does not have transgenic information embedded in it, any labels, we should be able to label them externally using some instruments that charge up. So they're like electrostatically driven. So if your tissue is very thick, then you put it in a chamber that there are some charges, and they push your marker inside through such tissues, which are embedded within agros. So within the Center for Brain Health, we also have some instruments that could enable that.
Oh, that's wild. Thanks for answering.
No worries, yeah, excited.
So I will point out that Mehwish did hold, I think, the first brain tissue clearing workshop in maybe all of Canada here within a year or so of her coming out. But I do think it might be a really interesting opportunity to get some of the clinical pathologists together, especially from non-neurological backgrounds, to kind of put our heads together and what could be the priorities, at a department level to kind of build up this capacity and take advantage of these really emerging technological advances.
Absolutely. We actually have a workshop coming again this year on May 2nd, and I'd be happy to share the details, where we are also bringing in one of the giants who started this work in US, who will be giving a talk, and there'll be a showcase of what has been done on UBC in the last 2 years. So happy to also add some sort of discussions around that and organize something.
Are there any other questions? I don't see anything in the chat, but please do go ahead if anybody from the audience has any other questions.
Could I just ask you, Mehwish, to put your email in the chat? Because I'd like to loop you in with someone possibly. Thanks.
Absolutely. There you go. It's my first name and second name with ubc.ca.
Super. Well, again, thanks to everybody for listening. It's been such a pleasure to mentor Mehwish. All of this work is hers. Basically, I was able to give her an opportunity to come into the lab and work with our Chimera model, but all of this stuff is 100% her doing. And so I think it's very clear she has an incredibly bright future ahead of her. And I think if we can extend Mehwish's network to include more members of the department, especially in non-neurological areas, that would really deepen the her opportunities and scope of work that she could be really engaged in. So welcome anybody to interact with Mehwish. As you can see, she's a wonderful teacher and a mentor and breaks things down. So they're so accessible. Even I can understand a little bit about the magic of what she's doing. David, thank you for the opportunity to have Mehwish present today.
Yeah, we also, as well as Jonathan, Eric McGuinness is on the call, who's like a heavy hitter in anatomic pathology across multiple organ systems and may have some ideas as well. And I just want to, since I saw him, he's there, wanted to make those introductions, and I'm sure he was as excited as I am as well about this.
Fantastic.
Okay, I think that's it then for today. I wish everybody a wonderful weekend.
Thank you so much for listening. Thank you, David. Thank you, Cheryl. Bye.
Bye-bye.